Good morning, everyone. Thanks for participating in our symposia, Phenotypic Still Matter in the Genomic Era. Today, I'm glad to introduce our next speaker, Dr. João Pedro, also known as GP Fontenelle. Dr. Fontenelle is currently a postdoc fellow at the University of Toronto, and he's working there in population and landscape genetics of cichlid eruptive species. He's interested in how spatial and temporal ecoevolutionary dynamics affect micro and macro evolution, and in the use of integrative approaches using phenotypic and genotypic data to understand biogeography and phylogenetic relationships of neotropical freshwater stingrays. He will present his talk in English and will answer questions in Spanish, Portuguese, or English. Welcome, Dr. GP, and you may start right now.
Hey guys, good morning. I hope you can all hear me. Thank you very much for joining us this morning here for this part of the conference. I am going to be talking a little bit about how interpretations of phenotype can affect our interpretations of molecular patterns in fast evolving, complicated, morphologically complicated groups. And I'll be using the Neotropical freshwater stingrays as an example. So before we get started, we just have to remember real quick that the relationship between phenotype and evolution is it's very intimate. So if we go back to Darwin, if we go back to the beginning of evolutionary theory, most of the questions that have been driving that is just, why do we see this diversity? Why do we see animals with different shapes, with different habits, working in different environments? What explain those differences? What are causing those differences? As we progress in the field, we now know that the phenotype, which is the observable physical properties of an organism, they are intimately related to the genetic composition of those lineages. So the manifestation of the genotype will be the phenotype under many environmental influences, all right? So if we want to be very simple, very straightforward, we can say that the phenotype is a direct result of the evolutionary process of each lineage. Phenotype is also very important when you're considering classification of the organisms, and especially when you're talking about the science of taxonomy. Taxonomy, where we're classifying organisms based on a discrete set of characters. We're trying to organize the living things and make sense of what is closer to what and what we are seeing. And even if you come from Aristotle, that was trying to discuss the hierarchy of animals, to Linnaeus that proposed the binomial system of classification that we still use today. Those classifications have been based on, and still are most of the time, based on phenotype, right? So it's something that we cannot neglect. What is really cool about when we are understanding the relationship between organisms and their shared characteristics is that we can come up with hypotheses of evolution, hypotheses of relationships, as we know as phylogenies, right? These phylogenies, they're based on similarities and differences in physical and genetic characteristics of the organism. So we can use what they have in common, what they don't have in common, and we can with the combination of those characters or information that each character will give us, we're able to come up and provide a hypothesis of evolution and classification of these organisms. So I've been talking a little bit about morphology and how it's important and how we use it a lot. But sometimes when you are coming with this hypothesis of relationship with these phylogenies, morphology sometimes do not provide enough information that will allow us to resolve a phylogeny. And when I mean resolve a phylogeny here is to come with relationship hypotheses that are not clear. So for example, if you look in this figure here from Vari 1989, that clade containing Curimatella, Siphocarex, Pseudocurimata, and Stingrays, we don't have a clear hypothesis on what is more closely related to what. And that might be related to morphology and phenotype, as we have a incomplete data set, and I'm going to say incomplete here in between quotation marks. It's just that we're still missing, we still haven't found those morphological characters, those phenotypical characters that will allow us to resolve that phylogeny. But sometimes we also have a phenomenon called convergence going on, when we have similar selective pressures that are not the same, producing similar phenotypes, but coming from different evolutionary origins, and that will cloud our phylogenetic signal for a little bit. So what we can do, the same way that we can look for more characters or more morphological characters to resolve that phylogeny, we can also increase our data sets by adding genetic data. And then we walk in, especially in the last decade of the 20th century, where we have morphology-based systematics and molecular-based systematics. Morphology-based systematics, more traditional way of approach, require group specialists and people that have been studying these groups for a long time. It requires a lot of training to be able to interpret that phenotypical variation. Availability of material, museum collections, those become fundamental for good works in this area. However, when we look at molecular-based systematics, we have a much faster process. We can automate that. We use a lot of computers. It's much easier to share sequences around. It's much easier to increase our sample size. Carrying tissues around is easier than carrying a 50 kilo stingray around. And because of that, it has many direct applications in other fields that we're going to be talking a little bit later on. When we walked into this molecular systematics-based era, phenotype, we come up with this concept of operational taxonomic units. An operational taxonomic unit is also based on similarity, but may or may not be equivalent to our taxonomic units defined by phenotype, may not be equivalent to a species, for example. OTUs are usually defined by genetic similarity, and how we relate OTUs to species or to genera, to those phenotypically defined units, will heavily impact our interpretation of our results, or even the results of some analysis, as we will see in a bit. So far, so good, great, we're increasing our data sets, but what happens when we have incongruences in phylogenies, in topologies of phylogenies between molecular and morphology data sets? If you've been working with this for a little while, you know that there is this debate in the field, both sides, people that work with morphology only, people that work with molecular data only. We have this kind of field going on. We have people arguing that both sides do bad taxonomy. They don't know what they're talking about. They're using wrong names. They're naming things in a wrong way. They don't have the capacity to identify properly what's going on. Some argue that there's limitations in morphology, especially regarding convergence and those missing characters. Some people say that molecular is looking at things too simplistically, that evolution is not like that. We know that genome evolves in different rates. They have different selections. They're prone to sequencing errors, to computer errors, to incomplete data sets. So what do we do from here? What happens? And that's how I'm going to talk a little bit about my group of study. So we have the Neotropical freshwater stingrays of the subfamily Potamotrygoninae, which is a marine derived lineage. What that means is that the most closely related lineage to this freshwater clade has a marine distribution. We hypothesize that these guys, the freshwater stingrays, have been evolving and diversifying in freshwater environments in South America for 20 to 25 million years, and we have around 42 species in 4 genera today. These fish, they're very famous for their colors and shapes. We have stingrays that have adult sizes around 25 to 30 centimeters. We have other species that the adult size is around 1.5 meters. We have different feeding behaviors. We have different niches that they occupy. but these guys are most famous because of their very vibrant dorsal color patterns, their phenotypes, which make them very famous in the aquarium culture, but also about the stings, and I'm pretty sure their stings, and I'm pretty sure if someone has dealt with these guys or spoken with people that deal with these guys, either fishermen or scientists, there's also a story, there's also a concern about accidents with those stings. And that's really cool until we try to understand how the morphology, how the phenotypes in this group evolve. These guys are very famous for inter and intra-specific phenotypic variability. And here I'm using an example of one species, Potamotrygoninae, that is found throughout the whole Amazon basin and Orinoco basin in South America. And we have this myriad of forms and colors and phenotypes that follow under the same taxonomic umbrella. To make that a little bit more complicated, we still don't have a species level phylogeny based on morphology. And what does that mean is that we have no hypothesis of phenotypic character evolution. So we don't know how characters are changing in the evolutionary history of that family, including the synapomorphy that we have for this group. So what do we see when we look from a molecular perspective? The most recent phylogeny that we have 38 out of those 42 species. We have many terminals from different localities. And what we see is this big plate here that's called the Potamotrygon core plate or core Potamotrygon plate in the genus Potamotrygon. That plate alone has 24 species out of those 42 that present a genetic diversity of only 2%. And if you're dealing with this for a little bit, you know that 2% is what many authors consider variation for one single species. We have a lot of incongruences between the nominal species based on phenotype and those molecular lineages. Out of those 24, 14 species are recovered as non-monophyletic. And just as an example here, I have Potamotrygon motoro in red, Potamotrygon erbignii in blue, and Potamotrygon scobina in green. And we can see that they're found all over that phylogeny, not recovering a monophyletic pattern. So what might be going on here? Do we have a problem with cryptic diversity? Was morphology is failing us? We still haven't found characters that would allow us to classify these guys based on phenotype alone. Or do we have markers, we have genes that are not fully coalesced and that doesn't reflect the real species tree, so that would be a problem with the molecular approach. Or we just have we have introgression that would cloud that molecular signal because we're having an admixture scenario of genomes going on. The same way we had the problem with phenotype that we increased our data set by adding molecular data, we can increase our molecular data using high throughput sequencing techniques, which are what we call genomics or phylogenomics, that increases our molecular data set in a lot of times. We'll talk about that in the next slide. Having a greater representation of genome would allow us, in theory, to address problems of having genes that are not fully coalesced. And with that, it will allow us to ask more complex evolutionary questions. So the Stingrays in the genomic era, this is what we have so far. We have generated a phylogeny, with 18,000 loci compared to the 4 ones that we have in that first one. And most of those taxonomic inconsistencies between phenotype and molecular lineage are resolved. Maybe. So the first thing that comes to our attention is that we have those same species under the phenotypic definition recovered in more than one lineage. That would be a cool evidence of cryptic diversity, where that morphology failed us. We're still missing those characters that will tell them apart. We have a scenario of convergent phenotypes, all good. We could also argue that we have incomplete lineage sorting, where genes haven't fully coalesced, so we have that clouded phylogenetic signal, and we can come to the conclusion that we have an underestimated diversity because phenotypes are not good. However, with the same data set, we find some lineages that are composed of 4, 5, 6 species with clear different phenotypes, as you can see here in the Potamotrygoninae complex, but the genetic differences that we see inside that lineage, they're not significant. So based on that assumption, what do we have here? We have a highly variable phenotype inside one species, and what we see is intraspecific variation. And then our conclusion now is that we are overestimating diversity. So depending on how we interpret these variations in phenotype, we will impact on how we interpret those genomic lineages that we recover, and we would have something between 30 to 50 species. But let's say that what we're seeing is introgression. Those phenotypes are good, those names are good, those lineages are good. How can we test for that? We can go for more in-depth genomic approaches and tests here. We can test for hybridization, incomplete lineage sorting. We can build up species tree, which allow us to explore the origin and genomic composition of those lineages. However, all of these analyses will require a taxonomic prior that is based most of the time in our phenotypic perspective of this group on how we define species or how we define units based on phenotype. Depending on our input on this kind of analysis, our results will be different. Even when we look at genetic composition and population structure, we still have to be worried about that. Those analyses rely heavily on a priori identification, mostly because these approaches were initially designed for intraspecific analysis. So we should be, in theory, working inside a single species. And why is that? Because most of these analyses rely on genetic variability within a lineage and in between lineages. So we're looking how much difference is enough for these programs to classify them into different things. And by including a different species or more than one species in that analysis, that bias the interpretation of variation that is allowed. So after this kind of like brief overview of the problems and the questions that we have to ask, is phenotype relevant in the genomic era? I would say that's more relevant than ever. I would say that we have to be really careful and we have to take our time and think a little bit on how we interpret that, especially because our input can bias our results, can bias our interpretations of micro and macro evolutionary questions. When we even unfold that to studies of diversification and phenotypic evolution, we still have to rely on solid phenotypic interpretations of those units. When we use barcoding and meta-barcoding, which are very important for conservation and management methods, those techniques, they rely on a solid curated database that will compare our samples to some voucher samples. And those guys are defined by phenotypic classification of species, for example. Conservation units, management efforts take into account species name. So it's very different from us to say we have one species that is endemic to one river, or we have one species that is found in that river, plus the whole Amazon basin, and that really impacts on how we unfold those efforts. Okay, so I've been talking a lot, for a fairly long time. I hope I got you a nice message across here. I have a lot of people to acknowledge, funding agencies, people that have helped me and helped us throughout these years. Special thanks for SBE for inviting me to talk here today. It's an honor to be part of this excellent group of people. And with that, I would like to thank everyone for showing up and listening to me. If you have questions, I am more than happy to answer them. And you can reach me out through email, through my website, or through Twitter. Thank you very much. That's it.
Thank you so much. You can keep your presentation if you want if any people want to ask you something. But yeah, we have a question right here.
Okay.
It is from Flavia. And she said, "Hey, JP, I was on work first. Were you able to morphologically identify each and every molecular sample is composed?"
I did my best effort to morphologically identify these guys. I've been working with this group for a while, but it's not very easy for those reasons that I said, that we have a lot of more intra and interspecific variation going on. Some of the descriptions of the species that we have from these guys are very old and very vague. So there is that kind of problem going on. And I thought that by using molecular data, that will help us to identify or this in a better way, until we start facing those problems that do we have one or more than one, and what the genomic differences mean. So yes, I tried, I did my best effort, and I still think there's a lot of work to be done with the morphology and phenotype of these guys.
Great. And there is more inside that question is,
okay,
then if you think about the barriers that interrupted the gene flow between ancient lineages, is it possible these stingrays got adapted to new environments and now they are able to cross these barriers?
that is an excellent question. That is something that we are working right now in the genomic paper. We're trying to interpret from a macroevolutionary perspective what's going on with the gene flow. I would say, and this is one assumption, I wouldn't write that in stone. I would say that some species, yes, they will be able to move across, and you see that with widespread species, while some species don't. I think there might be a relationship with ecological pressures there, sometimes competition, sometimes niche availability. But and I unfortunately do not have a definite answer for that yet. What I think that might be having is that we do have speciation going on with some sort of gene flow, and we do have evidence of that happening earlier in the evolutionary story of that group, which is pretty cool. So yeah, maybe I'll be able to address that question a little bit better in a few months.
And the last question is of this large question is, what are some hypotheses that could explain their hybridization?
I think the first thing that comes to mind here is if you read a little bit about evolution of elasmobranchs, which is the kind of the stingrays that we're working on, and teleos species, is that they say that the evolution of elasmobranchs happens on a slower pace compared to the teleos counterpart. So I would say that the first hypothesis, the most like out there hypothesis, is that because this evolutionary process is a little bit slow, the reproductive isolation between those lineages is not complete yet. So we would be able to see hybridization in the wild that would increase genetic diversity, and with that would be accelerating diversification and accelerating the generation of these phenotypic patterns, which is kind of counterintuitive when we think about what gene flow means. But we've been seeing that with some studies that have been coming in the past 2 years, that by increasing that genetic diversity by introgression, by hybridization, you're actually allowing for more opportunities of adaptation to changing environments. So I would say that's one hypothesis that also, of course, has to be tested, but could be one of those.
Great. Next question is from Carla Suarez. And she said, "Hey, JP, great talk. Did you try to combine morphological and DNA characters to produce your phylogenetic hypothesis? Are you considering other characters than dorsal color patterns?"
Hi, Carla. How are you? No, I haven't. I have not tried to interpret the morphological characters yet. I've been busy dealing with this very complicated morphological data, molecular data, and trying to understand those molecular patterns. I think that would be the next step. I think that the moment that we can produce a nice phylogeny that we think like, okay, we're going here with a solid species level relationship hypothesis, then would be the follow-up, the logical follow-up would be trying to bring morphological characters in so we can try to understand their morphological evolution. But yeah, not yet, unfortunately. Maybe we can collaborate in the future. That would be great.
Great. Next, it is from Vivian Amorim. Are there many software to test hybridization? Do you have some recommendations?
Um, Testing for the hybridization it's a little bit complicated. There is I would say the textbook way of doing it. You would have to know which one is the parental lineage, which one is the other parental lineage, and then you would see this is P1 and P2, ancestral genotype, and you would find the combination, which is a little bit hard, especially when we're dealing with this complicated group that you don't know what has coming from where, and we don't have a hypothesis what might be the hybrid, right? One way to do it would be to try to identify the limits of each species or each population, and then we see if you have something that fall in between that would show up as a mixture of those populations that we're interested in. But that is very complicated. As I mentioned, we need to be very sure of what is what, and phenotype plays a bigger role. What I would suggest is read a little bit about D-stats is a little bit of, or D-stat tests, because you can do that to test for hybridization and ILS. It's that big square that I showed that you have like the pink and the blue. So one that I would suggest is D-Suite, D-S-U-I-T-E. Google it up, maybe it will help you with your questions.
Great. Next is from William Ribeiro. Excelente apresentação, tendo em vista o reconhecimento de espécies críticas, como produzir guia de identificação para serem utilizados por ecólogos, biólogos de conservação, etc.
Oi, William, tudo bom? Cara, essa é uma excelente pergunta. Eu acho que até vai até um pouco mais para o aspecto teórico, né, do que é uma espécie e o que não é. Se a gente for para Linneu, a gente usa o termo espécie como a menor unidade que a gente consegue identificar taxonomicamente, né? Então, é complicado. Eu acho que, por estar lidando com esse grupo, é um grupo que a gente ainda tem muitas perguntas, né, muitas incertezas. Eu acho que usar a distribuição é uma coisa que pode ajudar bastante, né? Se a gente até vai para a área de ornitologia, de masto, o pessoal usa isso, né? Você tem as variantes regionais. Eu acho que é um começo até a gente conseguir identificar aqueles caracteres que vão definir se é espécie 1, espécie 2, espécie 3. Mas eu acho que faz parte do processo, infelizmente, ou felizmente, que é bom que tem trabalho para bastante coisa para fazer ainda. A gente ainda está engatinhando e, eventualmente, eu espero que a gente tenha um dataset de caracteres que possam ser usados para identificação em campo.
Muito bem. So next is, is from Angela Amarillo. Do you see phenotypic plasticity in this group? How can it be interacting with genetic variability?
Hi, Angela. That is a really good question. If I'm being completely honest, I have not got to that point yet. I haven't tried to work with phenotypic plasticity for this group, mostly because there's a lot of questions regarding the ecology of these guys. We know that there is some relation between feeding behaviors and morphology, especially of the jaws, but there's still a lot of incomplete data sets about feeding for these guys. We're not completely sure on what they eat, on what they have as an ecological niche that I think are very important when you're addressing phenotypic plasticity, right? So I don't know. Actually, that is a really good question. Hopefully we'll be able to answer that in the future for sure.
Great. We have 3 more questions, and I think that we can answer them. Carolina Machado. Ótima apresentação, JP. As sinapomorfias que suportam as espécies, elas estão bem delimitadas? Eu pergunto porque uma das espécies possui um padrão de coloração muito diferente, né? Provavelmente sua hipótese a priori de identidade pode não ser a melhor, uma vez que a morfologia é muito complicada. O que você acha disso?
Oi, Carol, tudo bom? É uma boa pergunta. E aí eu acho que tem um problema que a gente que está trabalhando com essas raias, a gente ainda não consegue resolver. A gente não tem, para a maioria das espécies, sinapomorfias a nível de espécie, né? O que eu mencionei, a gente tem a sinapomorfia para subfamília. A gente tem algumas variações morfológicas que se aplicam a 1 ou 2 ou 3 espécies ao mesmo tempo. E como a gente ainda não tem essa hipótese de evolução morfológica, fica muito difícil de interpretar esses caracteres, né? Eu acho que o ponto que você levantou é extremamente válido, é extremamente importante, que a gente precisa conseguir definir bem as espécies para conseguir entender os caracteres, mas infelizmente a gente ainda não chegou. Eu espero que com esses dados de genômica, pelo menos, a gente consiga ter uma ideia melhor da diversidade desse grupo, de como essas espécies estão relacionadas, ou essas espécies nominais estão relacionadas, quantas espécies tem aqui que não foram identificadas ainda, e a partir daí a gente consiga investigar melhor o relacionamento entre esses caracteres morfológicos.
Beleza. A seguinte pergunta é de Laura Martinez. Have you ever found hybrids in the field? Is there any population genetic studies in some species?
There are a few population genetic studies that came out in the past 5-10 years about the especially using karyotype that signal for hybridization with these guys. The hybridization of different species of Potamotrygoninae is very common in the aquarium trade, so you can buy hybrids between species, so we know that they are biologically possible to hybridize. When I started dealing with molecular data sets, those are the evidences that started coming up. So like, okay, so we might have been having hybridization in the wild. There is no reason to see that there's no reason to think that they would not be able to hybridize in the wild, because we see that in aquarium. But hopefully by when we start publishing and advancing with this molecular studies, we'll provide better evidence of hybridization in the wild. But if you ask my personal opinion, and let's put this off record because it still have to be tested, I have a hunch that yes, some hybridization in the wild is possible, and that might be one of the causes of that great variability of patterns that we see, and even why some specimens, when you get them fresh, you see that they look like 2 species, and it's very hard for us to put them in one box, species A or species B.
Great. And then we have our last question. Our last question, that it is from Gilberto Bergamo. Que boa apresentação, João. Eu sei que você já tem muitos dados morfológicos e moleculares para interpretar, mas já pensou em usar outros dados ecológicos, por exemplo, para uma aproximação integrativa talvez para ajudar a resolver isso.
Fala,
Abraço.
Gilberto, tudo bom? Sim, eu colaboro com duas pessoas, o Dr. Matt Coleman, principalmente, que ele está muito interessado nessa parte da ecologia dos bichos, principalmente com a alimentação e diferenças fenotípicas quanto a isso. A gente também colabora com o Dr. Devin Bloom, que gosta de ver essas diferenças fenotípicas com mudanças de habitat, né? Transição de habitat. Então, sim, é uma coisa que a gente está pensando, só que pelas limitações que a gente ainda tem para definir o que é uma espécie, o que é outra espécie, qual é a variação dentro dessa espécie, é uma coisa que está bem engatinhando ainda, mas com certeza é uma coisa que é importante, principalmente se a gente levar em conta questões de diversificação e nicho, que aí essa parte ecológica ela é bem delimitante ali, né? Mas com certeza, espero que aí nos próximos anos a gente consiga desenvolver isso aí um pouco mais a fundo.
Beleza, essa era a última pergunta. Tem alguns comentários te parabenizando aqui, falando que teu trabalho é ótimo. E muito obrigado por participar, Dr. JP. Thanks for coming and share your amazing work with us. I think that's all. We can close now and see you in the next Zoom room.
All right, guys. Thank you very much. If you have extra questions, send me an email or a message. Thank you very much, guys.
Thank you. See
Cheers.
you.